Photosynthesis
by Sarah,
Katie,
Megan,
Sandia.
The leaf is a very intricate place. We have the guard cell, which lets carbon dioxide and oxygen in and out of the cell, mesophyll photosynthetic cells, airspace, waxy cuticle, upper epidermis, palisade mesophyll, spongy mesophyll, vein, vascular bundle, lower epidermis, and waxy cuticle. And of course, we have the stoma.
The stoma is where most of photosynthesis takes place and where we will be focusing in teaching you all about the processes of photosynthesis. The photosynthesis reaction starts with carbon dioxide, water, and energy and results in oxygen and glucose. Though the equation may seem intimidating at first, we can break it down into two reactions, the light reaction and the dark reaction. The light reaction starts with light and water and results in oxygen, and the dark reaction adds carbon dioxide and results in glucose.
Now on to the light reaction.
The light reaction begins with H2O, NADP, and ADP, and results in H positive plus O2, NADPH, and ATP.
Light-dependent reactions take place in the thylakoid membrane. The outside of the membrane is called the stroma in the chloroplasts.
The reaction starts with light, a P680 photon hitting a chlorophyll pigment molecule. This pigment molecule absorbs the light energy and transfers the energy via electrons from chlorophyll to chlorophyll molecule. The electron transport chain begins. The chlorophyll electrons reach the reaction site and break apart water in oxidation into 2H plus and O2. Oxygen is the product here. This is considered the PS2 stage. Another photon of light energy hits the PS1 protein, changing NAD plus into NADPH. The H plus from PSI2 is still on the inside of the thylakoid membrane, the thylakoid lumen, and goes through ATP synthase, creating the energy needed to bond ADP with P, but resulting in H plus and ATP. So in summary, light hits protons in the thylakoid membrane, splitting H2O into 2H plus and O2, taking energy from NADP to create NADPH and ADP to create ATP.
Dark reaction!
light independent reaction, or also known as dark reaction. So it first starts out with H plus, and then plus 6 CO2 plus ATP.
Pow.
And it produces glucose, which is 6
C6H12O6.
Which is glucose.
plus RUBP. Woohoo!
Phase 1 of the Calvin cycle in the light-independent reaction converts 3 CO2 into 6 3-PGAs, which is phosphoglycerates. Phase 2 is the reduction of 3 phosphoglycerates to G3P. In first, 6 ATP is converted to 6 ADP and 6P. Then 6 NADPH is converted to 6 NADP. Phase 2, the reduction of 3 phosphoglycerate to G3P. First, ATP is changed to ADP and P. Then 6 NADPH is converted to NADP and H plus. Finally, the 6 G3P is changed to G3P. Phase 3, the regeneration of RUBP from G3P. First, the 5 G3P left over from phase 2 is taken. Then 3 ribulose and 5 phosphate. And then 6 ATP is changed into 6 ADP and 6P. That's the end of the Calvin cycle, folks.
Also, fun fact, guys, the Calvin cycle gives off two products, 3 RUBPs and G3P, which is sucrose or starch.
Cam plants. Some plants perform photosynthesis in a slightly different way, and these are called cam plants. Cam plants are plants that are adapted to hot and dry environments. A couple examples of these plants are cacti and pineapples. In cam plants, the initial CO2 fixation and the Calvin cycle processes are separated between night and day, whereas other plants divide photosynthesis stages by different sections in its cells. The stomata of cam plants open at night, when the temperature is lower and the humidity is higher. This allows the plant to reduce the water loss from its leaves.
Now back to the anatomical leaf diagram. Did you know that C4 plants are plants that undergo a specialized extrametabolic pathway? CO2 is also transferred from the mesophyll cells
Which is right here.
to bundle sheath cells
Which is here.
by a pump. CO2 gets dissolved, forming bicarbonate.
The C4 pathway is an adaptation from natural selection, allowing less photorespiration. C4 pathways ensure Rubisco encounters high amounts of CO2, meaning it is unlikely to bind to O2.
Some examples of C4 plants are grasses, corn, and sugarcane. C4 plants can be found in hot, dry conditions, which causes CO2 to not get into cells from closed stomata. There is excess carbon, and also cells form
In
oxalate.
bundle shell cells, Rubisco attaches to oxygen. Only one PGA molecule forms with a glycolate molecule and degrades to CO2. 4-carbon oxoacetate. Therefore, the name C4.
C4 plants divide into mesophyll and bundle sheath cells. 2 steps in mesophyll cells include light-dependent reactions and preliminary fixation of CO2, which are molecules called malate.
CO2 is released from malate in bundle sheath cells, where it is fixed by Rubisco. PEP is then recycled back into mesophyll cells and carbohydrate products of photosynthesis, which are distributed through plants. CO2 creates a higher concentration of CO2.
C4 plants have a lower light condensation point and a higher light saturation point.
The end!